So this is going to physically spin in the membrane like that. And that kinetic energy is used to pick up a phosphate and an ADP and stick them together, create a very high energy covalent bond. Okay, so question about this process so far. Has there's a difference, right, between active and passive transport. Passive transport doesn't require energy. It's just the following of molecules across the diffusion, or with the diffusion gradient. Active transport actually requires some energy. So where do we have active transport occurring, and where do we have passive transport occurring?
Active transport is from the proton going through the chlorophyll molecules.
Right here?
Yeah, and then down, and then it goes to the ADP synthase.
And is that active or passive?
It's active.
Passive. So this is a really interesting system, right? Because we have active transportation occurring right here. And the energy that we're using is this electron. So we have active transport at the point of the electron transport chain. But then we have the passive movement of protons from a high concentration to a low concentration that's facilitating the manufacture of ATP. So active transport creates a proton gradient. Passive transport creates ATP. I'll say that again. Active transport creates a proton gradient. Passive transport creates ATP. Now I'm going to give that passive transport a specific name, and this is a name that's going to be true for photosynthesis and also for cellular respiration. And that name is chemiosmosis.
That's for passive transport?
That's for the passive transport used by ATP synthase to create ATP.
What was it? Active transport creates what?
Active transport creates the proton gradient. Passive transport creates ATP. And that passive transport is called chemiosmosis.
Is it called that because it creates a chemical reaction?
It's called that because it's an osmotic, a diffusion type process for a chemical, which is that proton. Okay, so that's photosystem 2, the first process within the light reactions. Questions about this so far? Yeah.
So in like the very first step with the photon, is that an active transport?
So that's really neither, because we're not moving something across a membrane.
Okay.
Right? We're just it's passive. We're not actively moving the energy.
Mm-hmm.
That energy is just kind of moving between chlorophyll molecules.
Okay.
Good question. Others? Okay, so let's move to photosystem 1. Now, the same sort of process occurs in photosystem 1 as in 2, right? So the first thing to happen with photosystem 1 is what? Photon. Yeah, we're going to get another photon. It comes along and strikes a chlorophyll molecule. And then it gets excited, and we know that excitation means that an electron in the magnesium ion moves out one energy shell, right? So that's excited, it moves up. And then when it's finished with that energy, that electron falls back to its ground state, and the energy that was represented by that increased distance between the nucleus and the electron is transferred over here to an adjacent chlorophyll molecule. And that energy is passed ultimately to P700. Right? We're okay with that so far? Okay, so P700 becomes so energized that what happens to it? Yeah, it gets oxidized, right? So it loses an electron up to the primary electron acceptor. Okay, so far the process is identical to that in photosystem 2. However, something different reduces P700 than reduced P680. What reduces P700? The electron from P680, right? So this electron that fell through the electron transport chain comes over here and it reduces P700. Okay, and then we get the same thing happening, right? We have this electron that was taken up by the primary electron acceptor, and it's passed over to the electron transport chain. And then that electron just falls down the electron transport chain like this, in a series of redox reactions. So what's the purpose of the electron transport chain here? Yeah, exactly right. It's the same thing. It's to create a proton gradient. So it's going to pass protons from this side of the membrane to the other side. And what's the source of those protons? Yeah, those that fell back through ATP synthase. Okay. You okay with this so far? All right, so we have a high proton concentration on this side of the membrane, low proton concentration on this side of the membrane. Now, last time I told you that really there are two fates for those protons. What are those two fates, do you recall? One ends up over here on NADP plus reductase, really on NADP plus. Where do the others go, potentially? Do you remember?
Sugar?
So some of them might be recycled back to go back through
Start
ATP
all over.
synthase again. Yeah. But we're just going to graph those or diagram those that are following the NADP plus reductase path. Okay, so with NADP plus reductase, what happens is we need to take NADP plus and we need to add to it an electron. And we need to add to that a proton. Yeah. Doesn't that seem weird, the way I wrote that? Because with electron, I have E for electron, negative charge, because electrons have a negative charge. Why for a proton did I write H? Because of the hydrogen, right. What about hydrogen ions make it a proton? And how many neutrons? 0. So when that hydrogen atom loses its electron, all that's left over is a proton, right? So we take NADP plus, add to it an electron and a proton, and we get NADPH. Exactly right. What's the purpose of NADPH? Yeah, it shuttles electrons around specifically, right? I mean, that's all it does. It's just carrying electrons from one spot to another. Now, something that you guys need to know about both NADPH and ATP is that they are incredibly unstable. You can't store NADPH or ATP. Once you make it, you have to use it really, really quickly. Otherwise, it just naturally degrades. And that energy or those electrons are lost. Okay. So, we know that these protons perhaps come over here and can be attached to NADP+, right? And that electron comes from where? From the bottom of the electron transport chain, right? So that's where we get that electron. So, what's the purpose of ATP? Why did we make that? It's energy, right? Okay, so we've made a ton of energy, and we've made a bunch of electrons in a transferable sort of format. So that's important because the second part that we haven't talked about yet, the second part of the photosynthesis reaction, is taking carbon dioxide and converting it or reducing it into sugar. So this is going to be a reduction reaction. We're going to reduce carbon dioxide into sugar. Is that an endergonic or an exergonic reaction?
Endergonic,
It's endergonic, and Linda, why do you say it's an endergonic?
because it's releasing into the, it's absorbing into the other.
Okay, so you're investing a bunch of energy from here and making something bigger, right?
Yep.
Okay. So since it's endergonic, that means that it inherently requires some energy, right? So that's why we use ATP. And it's a reduction reaction, so it's going to require some electrons. ATP drives the reaction, that's the energy used to drive that reaction, and NADPH supplies the electrons for the reaction.
Can you repeat that?
You betcha. ATP drives the reaction, it's the energy used in that reduction reaction, and NADPH supplies the electrons for that reduction reaction. Yeah.
So is that NADPH's job then?
That's all it
To just
does.
create electrons, or
To-
to supply
it-
electrons?
That's a better way to think about it. To take electrons from right here over to here. It just shuttles them.
It's not like create some, it's like kind of create some vesicle for the electrons.
It's just it's a carrier mechanism, kind of like a vesicle is on a larger scale.
So would it be fair to say then with the NADPH, all it does is create a transport for the electron?
That is very fair to say.
Okay.
Yeah, it's just transporting electrons about. Yeah.
So would this be correct? NADPH supplies electrons for the reduction of CO2 into sugar.
That would be correct. Yeah. Other questions? Okay, so then I have a question for you. Would you like to see this on the exam?
Yes.
Yes.
You'd like to diagram that?
Yep.
Okay. So be prepared to diagram that and answer some questions about it. Okay. I like it. Okay. So does anyone want, do you guys want to
Yeah.
take a picture of it?
Like I can memorize it by section.
Here on a picture board.
Let me send you this.
Huh? He has it on the file.
Yeah, I think I do have this posted in the Canvas shell
Yeah, you do.
as a PDF. So when we were talking about, in chapter 5, when we were talking about enzymatic reactions, I mentioned something about a biochemical pathway. And a biochemical pathway is just a sequential series of reactions, right? Where you're taking one product and converting it into another. And that's all the light reactions are. It's just a long biochemical pathway in which ultimately ATP is created and NADPH is created. Cool? Okay. So let's go through this in the slides really quickly. Okay. So I know we've already gone over this, but the first thing to happen in the light reactions, and it happens right here, right, is a chlorophyll molecule becomes excited when it receives a photon. So that's stage one, it's excitation. In the second stage, that energy absorbed from the photon is passed between adjacent chlorophyll molecules. And then ultimately, an electron is passed from P680 or P700, which receives that energy, right, up to the primary electron acceptor. So that molecule, either P680 or P700, is oxidized and it loses an electron, and that occurs right here. To reduce P680, we split a water molecule. To reduce P700, we take the electron that was released from P680 and stick it back onto P700. So the electron, right, that's liberated from either P680 or P700 goes up to the primary electron acceptor, and then it's passed over to the electron transport chain, which creates a proton gradient. It creates, actively, a diffusion gradient. And then that diffusion gradient can be used for a variety of things. If it's used subsequent to PS2, it is used to create ATP, right? Those protons passively fall back through ATP synthase, and that passive energy creates a kinetic motion, kinetic energy within ATP synthase, which is used to create ATP. After PS2, it can either be used to create more ATP, or some of those protons can be used in the reduction reaction of NADP plus into NADPH. Okay, so ultimately, what we're making from the light reactions, and you guys, the purpose of the light reactions is to create ATP and NADPH. We're creating the stuff